Welcome to the Alabama Water Watch instructional video series. This video will show the proper method for collecting water samples used to determine the number of bacteria in the water body. This is the first step in a three-step process. The second and third steps, plating the samples and determining results, are covered in their own separate videos. For each site being tested, you will need a cooler with ice, 3 media bottles at air temperature, a sterile pipette, and a thermometer. Label your media bottles by site and replicate. If you sample multiple sites, be sure to use separate data sheets or keep record of monitoring site and bottle numbers that are grouped in sets of 3 replicates. For example, if you have 2 sites, you can number the replicates at the first site as 1A, 1B, and 1C, and the replicates at the second site as 2A, 2B, and 2C. Upon arrival at the site, measure the air and water temperatures. For instruction on how to measure these temperatures, please watch the video called Water Chemistry, Air and Water Temperature. Once you are ready to sample, unwrap a sterile pipette from the bulb end and avoid touching the tip of the pipette with anything except the sample water. Fill the pipette with sample water. Be careful to not disturb the bottom of the stream. Carefully squeeze the bulb and expel excess water to the desired sample volume. 1 milliliter sample volume is recommended and is usually sufficient. Be sure to use the same volume for each of the 3 replicates and record volumes on data forms. Remove the cap from the media bottle and hold it without touching the inside. Squirt the sample water into the bottle of liquid media without placing your hand or face over the open bottle. Cap the bottle securely and place inside the cooler. Repeat these steps until all 3 media bottles are inoculated with water from the same spot. Keep in mind that media must be plated within 3 hours of collecting the sample and should remain on ice and out of direct sunlight at all times. Thanks for watching. If you have any comments or questions, give us a shout.